Okay guys, so we will move to the second part. أول شي بدي لكم pages not required in the book, so pages 526, 27 and 28 not required. So please open your book page 529 and we will start by the paragraph entitled The Role of Chromatin in Regulating Gene Transcription. As we learned in chapter 2 كلنا بنعرف إنه the eukaryotic chromosome consists of DNA complexed with histones and non-histones proteins. So إذا بتتذكروا guys أنا وياكم بأول part من chapter 18 قلت لكم we have different levels in eukaryotic cells where you can regulate the gene expression. We described the transcriptional level شفنا قبل how by the addition of activators or repressors we can regulate the transcription of genes, but we will move to the beginning of this إذا بدكم pathway, and we will focus more here on the role of chromatin in regulating the gene transcription. مثل ما قلت لكم قبل شوي إنه الeukaryotic chromosome is composed of DNA complexed with histones to form a structure known by nucleosomes. already حكينا عنهم اللي هن like or similar to beads on a string. Each bead or each nucleosome is composed of 8 histones, 2H2A, 2H2B, 2H3 and 2H4, wrapped by DNA. شفناه هون 1.65 times, form this structure known by nucleosome. We moved from 10 nanometer chromatin fiber to 30 nanometer chromatin fiber by the addition of the H1 histone protein to the linker DNA. And finally, we added the non-histones proteins, اللي هن examples of scaffold proteins, finally leading to the structure of eukaryotic chromosome. We have to note that the nucleosome organization, how chromatin are organized, how the nucleosome are organized, has a repressive effect on gene expression. لنرجع للstructure of 10 and 30 nanometer chromatin fibers. وقت نكون عم نحكي about the 30 nanometer chromatin fiber, شوفوا guys كيف الستركتشر بتكون. الستركتشر بتكون tightly packed, tightly coiled. In this structure the genes are inactive, transcriptionally inactive. However, when you convert the 30 nanometer into 10 nanometer chromatin fiber, شوفوا كيف الستركتشر صارت less coiled, uncoiled structure, loose chains. So the structures رح يكون بهالform رح يكون في عنا active genes because the DNA chains, the nucleic acid chains are uncoiled. بهالform the DNA is present in the open conformation, so the proteins, especially the RNA polymerase and the general transcription factors, can access more easily the DNA, can bind easily to the strands of the DNA and start the process of transcription. In addition guys, إذا بتتذكروا عم نحكي about the repression of gene activity by histones. بدي منكم تخلوا ببالكم إنه الهيستونز اللي هن part of the nucleosome structures. النيوكليوزومز form the chromatin. كل هول الستراكشر nucleosome organization in the chromatin, the structures of the histones has a repressive effect on a gene expression. So خلينا نشوف how the scientists discovered this repression of gene activity by histones or nucleosome organization in the chromatin. They performed an experiment in vitro. In vitro means outside the body. They introduced in 2 different tubes. In the first tube, they introduced a transcriptionally active genes, and in the second one, they introduced transcriptionally inactive genes. And they treated these 2 types of genes by a DNA degrading enzyme known by DNase 1. اوكي سو اخذنا 2 tubes، حطينا باول واحد transcriptionally active gene بتاني واحد transcriptionally inactive gene وحطينا بالاثنين DNA degrading enzyme. It's an enzyme that can degrade the DNA known by DNase 1. What they discovered that the DNase 1 is highly sensitive for the transcriptionally active gene. يعني this enzyme can degrade, can destroy easily the transcriptionally active genes. However, this enzyme is resistant أو the DNA اللي هي transcriptionally inactive genes are resistant to the DNase 1. معناتها this enzyme cannot cleave, cannot destroy easily the transcriptionally inactive gene. What we can conclude from this experiment? The scientists concluded that this increased in the DNase 1 sensitivity for the transcriptionally active genes does not mean that the DNA is not organized into nucleosome, but instead it means only that the chromosome is less coiled in this region, making the DNA more accessible to DNase 1. So إذا بتطلعوا معي على هاي الفيجر guys, let's consider this part represent the transcriptionally inactive gene لأنه نحن اتفقنا قبل إنه when the DNA is coiled tightly packed, the genes present in this region are transcriptionally inactive. So إذا أخذنا هيدا الريجن وحطينا معها the DNase 1, it's not easy to cut this region because the structure is tightly packed, tightly coiled. It's a tangled structure with knots إذا شايفينهم كأنه انعقد. However, when you open the conformation, when you create an uncoiled or less coiled structure forming the 10 nanometer chromatin fiber, صارت مفتوحة أكتر الستركتشر open conformation معناتها the DNase 1 can cut easily these region اللي هن DNA strands open conformation and more accessible to the enzyme اللي هي DNase 1. So, the scientists from this experiment discovered the presence of region known by hypersensitive sites. So هول الريجن اللي عم نشوفهم هون هن hypersensitive sites for the DNase 1 أو نسميهم hypersensitive regions. These sites or regions are the first to be cut with DNase 1. Where you can find usually this region inside the body, in vivo, inside the cell. These regions known by hypersensitive sites mainly are present in the region of the promoter just before the +1 start site where the transcription starts. So نحن أول step before the process of transcription, we need to uncoil the promoter region in order to make the DNA more accessible by the RNA polymerase and the general transcription factors in order to transcribe the DNA starting from +1. But it's not only the region of the promoter, but also you can find in the region of the enhancer كمان hypersensitive sites because activators should bind here and then recruit co-activators, the RNA polymerase, and the complex will bind to the general transcription factors at the level of the promoter and enhance the transcription 100-fold. We will move to this slide. إذا بتلاحظوا guys عم جرب أقرب لكم الفكرة شوي. إذا كان عندكم wool filaments خيطان صوف، إذا كانوا coiled مثل هالطريقة with knots, it's difficult to cut. Right? It's difficult to cut these strands. However, when you uncoil this wool filaments, when you open the conformation, when you create an untangled, without knots, so this structure is easy to cut or to destroy. معناتها بطريقة تانية when they performed the scientist experiment in vitro وقتها كانت ال DNA transcriptionally inactive. ليش ال DNase 1 اللي هي it's similar to a scissor here ليش ال DNase 1 cannot cut or destroy the transcriptionally inactive genes? The reason is إنه they are present in a tangled tightly packed coiled structure 30 nanometer chromatin fiber. However, in the second figure وقت نشوف in open conformation the 10 nanometer chromatin fiber, the scissor here representing the DNase 1 in vitro in the experiment قدرت قصت بسهولة كتير هيدا الفيلامنت. معناتها إنه we have an uncoiled structure when we are present in the case of transcriptionally active gene. So the DNA will be in open conformation. The DNA is easy to be accessed by the transcription machinery, and these proteins can bind and start the process of transcription at the +1 start site. رح نقطع على آخر part نعمل فيه like summary للي شفناهن هلا. So إذا بتلاحظوا guys, this is the 30 nanometer chromatin fiber tightly packed. هون قطعنا أكتر للnucleosome structure where you can find the chromatin 30 nanometer chromatin fiber where you can find the chromatin in less highly coiled structure. Here at this level, when you have an open chromatin, إذا لاحظتوا النيوكليوزوم تفككوا عن بعض الهيستونز they are not present. In this region إذا بناخد هيدا الريجن اللي هون ومنروح more إذا بنعمل زوم على الانتراكشن between the nucleotides بتشوفوا إنه the DNase 1 in this open chromatin can cut easily these sites. So حطيت لكم هون points to summarize اللي حكيتهم. The hypersensitive sites is a short region of chromatin. It's detected by its super sensitivity to cleavage by DNase 1 لأنه it's an open conformation. The nucleosomal structure is less compacted. Hypersensitive sites are found only in genes is being expressed and do not occur when the gene is inactive. كلنا بنعرف ليش لأنه when the gene is expressed معناتها it's present in the 10 nanometer. However, when the gene is inactive, is present in the 30 nanometer, more coiled, more packed. And the hypersensitive sites are generated as a result of the binding of transcription factors that displace histones octamers. Octamers means 8 histones present in the nucleosome structure. We will move to the second part of this video, and we will talk more about the facilitation of transcription activation by remodeling of chromatin. So نحن if our chromatin is present in the 30 nanometer, in order to actively transcribe the genes, we need to convert the 30 nanometer into 10 nanometer. How we can convert the 30 into 10? By a process known by chromatin remodeling. مثل ما عم تشوفوا هون chromatin remodeling بتحول ال 30 ل 10 and you can facilitate the transcription activation. In order to remodel the chromatin, we need two classes of proteins. رح نحكي عن أول class بهيدا slide وبنرجع later بنشوف class 2. The first class of protein involves enzymes that modify the nucleosome by either acetylating or deacetylating the core histones. So طلعوا معي guys على هيدا الفيجر. منبلش بال 30 nanometer chromatin fiber, it's a closed chromatin. By the addition of acetyl group, you will convert the closed chromatin into a relaxed chromatin, means 10 nanometer chromatin fiber, and you can activate the process of transcription. However, the opposite is a بدكن direction. The relaxed chromatin 10 nanometer by deacetylation عم بمشي بالعكس restore the 30 nanometer chromatin fiber وهيك بترجع the structure closed and the genes will be inactive. So خلوا ببالكم إنه in order to remodel the chromatin structure we need one class of protein involving enzymes that modify the nucleosome by acetylating or deacetylating the core histones. خلينا نبلش بأول طريقة. بدنا نمشي من 30 nanometer ل 10 nanometer. We have an enzyme known by HAT for histone acetyl transferase من اسمها. So it's an enzyme that add or transfer acetyl group to histones present in the nucleosome structure. But this enzyme was recently named CAT. Why CAT? For KAT, K lysine acetyl transferase. كلنا بنعرف إنه الأمينو acids are represented by letters. Here the letter K represents the lysine. So the same enzyme فيكن سموها HAT or CAT. CAT is more specific لأنه عم بتقلنا على أي amino acid I will add the acetyl group. Here it's the lysine residue of the histones. So إذا بنجي بنطلع guys, this is the structure of the lysine اللي هو one amino acid of the histones, core histones in the structure of the nucleosome. By the addition of acetyl group by CAT's enzyme, هيدا الأسيتيل جروب سكناه فوق. شوفوا شو بيصير بالstructure of the nucleosome. هاي كانت البيت اللي هي عبارة عن 8 histones, core histones wrapped by the DNA. مجرد ما زد لهم acetyl group, شوفوا شو صار guys. The acetyl group will neutralize the positive charge of the lysine. ما لازم ننسى إنه الهيستونز proteins are positively charged لأنه بيعملوا attraction للnegatively charged phosphate group of the DNA. So حطوا ببالكم إنه الهيستونز are positively charged. ليش هن positively charged؟ لأنه عندهم basic amino acid، يعني they are positively charged basic amino acid. One of this amino acid basic is the lysine. Lysine is positively charged بالهيستون. هي اللي رح تساعد الهيستون يلقط ال DNA to form the nucleosome structure. إذا نحن جينا حطينا acetyl group على الليزين, the acetylation of the lysine will neutralize the positive charge. معناتها الليزين بطلوا positively charged. الهيستونز بطلوا positively charged. معناتها no more attraction with the negatively charged phosphate group of the DNA. So what will happen برأيكم للnucleosome structure؟ بتضلها tightly packed أو برأيكم رح تصير loose, unpacked, uncoiled? The answer is by the addition of acetyl group, the structure will be uncoiled, unpacked structure. So with increasing the acetylation, the positively charged histones slowly lose affinity for the negatively charged DNA. And the 30 nanometer chromatin fiber loses histone H1 and changed conformation to a 10 nanometer chromatin fiber. Not only we will lose the histone H1 from the linker DNA, كمان بقلب النيوكليوزوم إذا بنحكي على الكور هيستونز بقلب الهيستونز بطلتوا رح تشوفوا إنه the nucleosome is tightly packed. كمان رح تشوفوها it's a loose structure, uncoiled. In this form, you will help the promoter إنه يصير uncoiled and it's more accessible for activation of transcription. هاي إذا عم نقطع guys من أول part اللي هي closed chromatin relaxed chromatin. بس إذا نحن خلصنا transcription وبدنا نرجع الستركتشر من relaxed ل closed chromatin، كل اللي لازم نعمله هو is to remove the acetyl group added by the HAT or CAT enzyme. In order to remove this acetyl group, we need another enzyme known by HDAC for histone deacetylase. This enzyme will remove the added acetyl group and restore the 30 nanometer chromatin fiber. We will move to the second class of proteins involved in the remodeling of chromatin. So it's the chromatin remodelers. The class 2 is an ATP-dependent nucleosome remodeling complex. So من اسمه, it's a complex that can remodel the nucleosome, but in the presence of ATP. So it's a large multi-protein complexes that remodel chromatin by using the energy of ATP hydrolysis. في شغلة لازم نحطها ببالنا، هيدا اللي بدي أقولها بتطبق على الكلاس 1 وعلى الكلاس 2. So أول وحدة اللي هن الانزيمز HAT, CAT or HDAC، وعلى تاني وحدة اللي كلاس 2 ال ATP remodelers. These two classes are similar بأي معنى؟ First, the activators will bind to an enhancer. Then recruit يا أما the class 1 remodelers or the class 2 remodelers. These two classes will alter the nucleosome, open the structure of the promoter or the sites before the +1 start site of the transcription, facilitating the binding of the transcription machinery to the core promoter. So فهمتوا كيف عم نمشي step by step. أول شي بيجوا الactivators بيقعدوا على enhancers. These activators will recruit the class 1 or class 2 of the class 2 of remodelers. يعني يا أما منجيب الهات أو منجيب ال ATP dependent nucleosome remodeling complexes. هول ال two classes بيجوا بيغيروا ال structure of the DNA at the level of the core promoter، بيحولوه من 30 nanometer to 10 nanometer chromatin fiber, less coiled, more relaxed conformation, open conformation. So the DNA in the core promoter now can bind to the transcription machinery and start the transcription at the +1 start site. In this slide, we will focus more on the class 2, how this class 2 will remodel the nucleosome. By one of these three is بدكن ways. The class 2 can slide مثل ما عم نشوف بأول وحدة can slide a nucleosome along the DNA, exposing sites for DNA binding protein. So هالclass of proteins remodelers بدهم يجربوا يحولوا the structure of the DNA from a closed conformation to relaxed open conformation. So شو بيعملوا؟ بأول طريقة this remodelers can slide، شايفينهم دفشوا can slide a nucleosome along the DNA. So صار في عنا DNA in a relaxed conformation. The DNA strand will be exposed for DNA binding proteins such as general transcription factors or RNA polymerase or activators. بطريقة تانية هول remodelers can restructure the nucleosome in place to facilitate the association of protein with DNA sequences. Or بالطريقة الثالثة these remodelers can transfer the nucleosome from one DNA molecule to another. So إذا لاحظتوا شلنا النيوكليوزوم structure from one DNA ونقلناها على another DNA molecule، وهيك بالfirst DNA molecule صار في عنا DNA strand, less coiled, open, relaxed conformation, ready to accept the transcription machinery and to start the process of transcription. So as a conclusion, please keep in mind, in order to start the transcription, first we need to convert the DNA from 30 nanometer closed conformation into 10 nanometer relaxed conformation. كيف بدنا نحولهم؟ في عنا واحدة من التو classes اللي فينا نستعملهم، يا أما class 1 by the addition of acetyl group to the lysine residues of histones, so it's an acetylation using CAT or HAT enzyme. وبتاني class في عنا in ATP dependent nucleosome remodeling complexes. These complexes depends on the energy produced by ATP hydrolysis to slide, to restructure, or to transfer the nucleosome from the DNA sites, creating a relaxed open conformation of the DNA, exposing the sites for DNA binding proteins.